In this mini tour, we'll build an intuitive picture of gene regulation terms from A to T and finish with a quick check: which mechanism uses repressor proteins. Let's start with a simple eukaryotic gene map. The orange block is the promoter. The purple complex TFIID sits on the TATA box, and the green enhancer can loop in from far away to boost transcription. The cyan bar below is a CPG island often found in active promoters. We'll highlight each: T promoter, A and Q, TATA box binding general transcription factor TFIID, B enhancer, and C CPG island. Quick definitions on screen. T promoter, a DNA region upstream of the transcription start site that directs initiation. A and Q, TFIID. Its TATA binding protein recognizes the TATA box, recruiting RNA polymerase II. B enhancer, a cis element that boosts promoter activity through looping. C CPG island, a CG-rich region usually unmethylated at active promoters. Epigenetic layers regulate access. D DN, a methyltransferase adds methyl groups to cytosines in CPG, typically repressing transcription. E histone variants change nucleosome properties. F chromatin remodeling slides or ejects nucleosomes to open or close DNA. D DNA methyltransferase adds methyl marks, represses. E histone variant, a non-standard histone that alters chromatin. F chromatin remodeling, ADP-dependent nucleosome repositioning. Now, a bacterial operon model. S an operon is a single promoter controlling several genes, producing J, a polycistronic mRNA with multiple ribosome binding sites. N negative control uses a repressor bound to the operator to block transcription. P attenuation can prematurely terminate transcription via RNA hairpins. S operon, one promoter, many genes. J polycistronic mRNA, one transcript encodes multiple proteins. N negative control, a repressor on the operator blocks RNA polymerase. P attenuation, RNA structure causes early termination. RNA can regulate itself. M a riboswitch in the 5' UTR binds a metabolite to turn translation on or off. G short interfering RNA guides the RISC complex to slice target mRNA for silencing. M riboswitch, ligand-bound RNA changes structure to control gene expression. G siRNA, dicer makes siRNA, which loads into our RISC to cleave matching mRNA. In eukaryotes, introns are removed by splicing. H a constitutive exon appears in every mature mRNA. K alternative splicing mixes and matches exons to create different isoforms. H constitutive exon, always included. K alternative splicing, different exon choices, different proteins. Signaling ties in as well. L CAMP is a second messenger that binds proteins to change activity. I an allosteric enzyme changes conformation when an effector binds away from the active site. L CAMP, a small cyclic nucleotide messenger. I allosteric enzyme, activity modulated by effector binding at a separate site. O a temperate phage can choose between the lytic path, killing the host, or the lysogenic path, integrating as a prophage. O temperate phage, lytic or lysogenic life cycles. R promoter bashing is a technique to find important promoter elements by deleting chunks and measuring activity. R promoter bashing, sequential deletions to map functional DNA. A quick recap of two more terms. A prime, constitutive gene, expressed continuously, not strongly regulated. T promoter, the DNA region where transcription begins, with motifs like TATA or -35 and -10. Final check: which mechanism utilizes repressor proteins to inhibit transcription? The answer is N negative control. A repressor binds an operator or silencer to block initiation. Great job! You've seen A through T with visuals, promoters and TFIID enhancers, and CPG islands, methylation, histone variants, and remodeling, operons, polycistronic M, our NA negative control and attenuation, riboswitches, and our NA interference, splicing choices, CAMP and allosteric, temperate phage, and promoter bashing.